This video is about phylogeny and ontogeny of immune system, cells of immune system, and this particular study note is developed by EPG Pathshala site, Government of India, MHRD. So I would like to express my sincere gratitude to the developing team of this study note. Thank you so much. Hematopoiesis, B cells, T cells, natural killer cells, monocytes, macrophages, dendritic cells, neutrophils, basophils, eosinophils, and mast cells. First, introduction. The response to pathogens is coordinated by the complicated interactions and activities of the large number of different cell types involved in the immune response. The innate immune response is the first line of defense and occurs soon after the exposure of pathogen. Innate immune response is carried out by phagocytic cells like neutrophils and macrophages, natural killer cells, NK cells, and granulocytes. The adaptive immune response include antigen-specific defense mechanisms and may take days to develop. Cell types with critical roles in adaptive immunity are antigen-presenting cells, including macrophages and dendritic cells. So, antigen-dependent stimulation of various cell types, including T cell, B cell, and macrophages, all play critical roles in host defense. The immune system has large collection of cells, not only lymphocytes but also phagocytes and their relatives. Some immune cells take on all comers, while others are trained on highly specific targets. The immune system stores just a few of each kind of different cells needed to recognize millions of possible enemies. When an antigen appears, those few matching cells multiply into a full-scale army. After their job is done, they fade away, leaving sentries behind to watch for future attacks. Hematopoiesis. The term hematopoiesis refers to the formation and development of the cells of the blood. Hematopoiesis is a continuous process throughout adulthood. It is estimated that the average human produces 3.7 into 10 raised to 11 blood cells per day. This process is regulated by complex mechanisms. Cell division and differentiation during hematopoiesis is balanced by apoptosis or programmed cell death. If apoptosis fails, a leukemic state can occur. Leukemia. In humans, this process begins in the yolk sac in the first week of embryonic development. By the third month of gestation, stem cells migrate to the fetal liver and then to the spleen. Between 3 to 7 months gestation, these two organs play a major hematopoietic role. Next, the bone marrow becomes the major hematopoietic organ, and hematopoiesis ceases in the liver and spleen. Every functional specialized mature blood cell is derived from a common stem cell. These stem cells are therefore pluripotent. These stem cells represent a self-renewing population of cells. These cells have the potential to differentiate and to become committed to a particular blood cell lineage. Pluripotent stem cells differentiate into two major pathways, lymphoid and myeloid. Stem cells then become progenitor cells for each type of mature blood cell. These cells have lost the capacity for self-renewal and are committed to a given cell lineage. The myeloid progenitors develop into the cells that respond early and non-specifically to infection. Myeloid progenitors. Neutrophils engulf bacteria upon contact and send out warning signals. Monocytes turn into macrophages in blood tissue and phagocytose foreign invaders. Granule-containing cells such as eosinophils attack parasites, while basophils release granules containing histamine and other allergy-related molecules. Lymphoid progenitors develop into lymphocytes. Lymphocytes respond later in infection. They mount specific attack involving antigen-presenting cells such as dendritic cells or macrophages. Progenitor commitment depends upon the acquisition responsiveness to certain growth factors. The particular microenvironment, hematopoiesis inducing microenvironment or HIM, within which the progenitor cell resides controls differentiation. The hematopoietic cells grow and mature on a meshwork of stromal cells, which are non-hematopoietic cells that support the growth and differentiation of the hematopoietic cells. The meshwork of stromal cells include fat cells, endothelial cells, fibroblasts, and macrophages. Some of the hematopoietic growth factors are colony stimulating factor or CSF, multi-lineage colony stimulating factor, multi-CSF or IL-3. granulocyte macrophage colony stimulating factor or GM-CSF, macrophage colony stimulating factor or MCSF, then granulocyte colony stimulating factor or GCSF. Erythropoietin induces terminal erythrocyte development and regulates RBC production. Erythropoietin induces terminal erythrocyte development and regulates RBC production. Interleukins IL-4, IL-5, IL-6, IL-7, IL-8 and IL-9. CSF act in a stepwise manner inducing proper maturation. IL-3 multi-CSF acts early, possibly even at the level of pluripotent stem cell to induce formation of the non-lymphoid organ cells, erythrocytes. CSF act in a stepwise manner inducing proper maturation. IL-3 multi-CSF acts early, possibly even at the level of pluripotent stem cell to induce formation of the non-lymphoid cells, erythrocytes, monocytes, granulocytes, neutrophils, eosinophils, basophil and megakaryocytes. GM-CSF acts at a slightly later stage, but it is also induces formation of all the non-lymphoid blood cells. MCSF and GCSF act still later to promote the formation of monocytes and granulocytic cells respectively. I would like to summarize this once again. Colony stimulating factor or CSF, multi-lineage colony stimulating factor, multi-CSF or IL-3, then granulocyte macrophage colony stimulating factor, GM-CSF, macrophage colony stimulating factor or MCSF, granulocyte colony stimulating factor or GCSF. Erythropoietin induces terminal erythrocyte development and regulates RBC production. Interleukin IL-4, IL-5, IL-6, IL-7, 8 and 9. IL-4 stimulates B progenitors, mast progenitors and basophil progenitors. IL-5 stimulates eosophil progenitor. IL-6 stimulates the myeloid stem cell. IL-7 induces the differentiation of lymphoid progenitor into B progenitor and T progenitor. Then IL-8 stimulates the neutrophil progenitor. IL-9 stimulates mast cell growth. Hematopoiesis in humans. This is a kind of big explanation. Wait a minute. I'll be slowly trying to interpret this. Bone marrow, common myeloid progenitors, multipotent hematopoietic stem cell, that is the hemocytoblast, multipotential hematopoietic stem cell, which divides into common myeloid progenitor and common lymphoid progenitor. Here we could see approximate scale information is provided. The morphological characteristics of hematopoietic cells are seen in is the same May Giemsa stain or May Grunwald Giemsa stain. Alternative names of certain cells are indicated between parentheses. Certain cells may have more than one characteristic appearance. In these cases, more than one representation of the same cell has been included. Together, the monocyte and the lymphocyte comprise the agranulocytes as opposed to the granulocytes, basophil, neutrophil and eosinophil that are produced during granulopoiesis. E stain for basophil, neutrophil and eosinophil respectively as in basophilic promyelocyte. For lymphocytes, the T and B are actual designations. The polychromatic erythrocyte, reticulocyte at the right shows its characteristic appearance when stained in with methylene blue or azure blue. The erythrocyte at the right is more accurate representation of its appearance in reality when viewed through a microscope. Other cells that arise from the monocyte, osteoclast, microglia, central nervous system, Langerhans cells, Kupffer cell, liver. For clarity, the T and B lymphocyte are split to better indicate that the plasma cell arises from the B cell. Note that there is no difference in the appearance of B and T cells unless specific staining is applied. First, common myeloid progenitor, megakaryoblast, promegakaryocyte, megakaryocyte, thrombocytes or platelets. Thrombopoiesis. Once again, common myeloid progenitor, megakaryoblast, promegakaryocyte, megakaryocyte, then thrombocytes or platelets, thrombopoiesis. Then proerythroblast, pronomoblast, basophilic erythroblast, polychromatic erythroblast, orthochromatic erythroblast or nomoblast, polychromatic erythrocyte, reticulocyte, erythrocyte, red blood cell, erythropoiesis. Next, myeloblast, B promyelocyte, B myelocyte, metamyelocyte, B band, basophil. Then N promyelocyte, N myelocyte, N metamyelocyte, N band, basophil. Neutrophil. Then E promyelocyte, E myelocyte, E metamyelocyte, E band and eosinophil. Then monoblast, promonocyte, create monocytopoiesis, macrophage, myeloid dendritic cell. On this side you could see common lymphoid progenitor, lymphoblast, prolymphocyte, natural killer cell, large granular lymphocyte, small lymphocyte, B lymphocyte, T lymphocyte, plasma cell, lymphopoiesis, lymphoid dendritic cell and myeloid dendritic cell is also there. So this shows the process of hematopoiesis. Cells of immune system, stem cell, lymphoid stem cell and myeloid progenitor. So lymphoid stem cell, lymphocytes, B cell progenitor, T cell progenitor, natural killer cell. B cell progenitor again give rise to plasma cell and memory cell. T cell progenitor give rise to Th cell and Tc cell, T helper cell and T cytotoxic T cells. Then myeloid progenitor, granulocytes, neutrophil, eosinophil, basophil, mast cell, monocyte. Monocyte again divides into dendritic cell and macrophages. Divides into in the sense give rise to dendritic cell and macrophage. Origin and differentiation of cells of the immune system. Then cells of the immune system in detail. B cells. B cells account for 10 to 15% of circulating lymphocytes. They are called B cells because they were first discovered to mature in the bursa of abrasives and birds. Humans do not have this organ and B cell maturation takes place in bone marrow. Therefore, the name of the cells remains as B cells. B cells circulate around the body in the bloodstream. When activated, they release huge amount of antibodies. The major function of B lymphocyte is to develop into antibody-secreting plasma cells after stimulation by antigens like bacteria, virus, tumor cells, etc. Antibodies are specialized protein molecules that specifically recognize and bind to specific antigens that cause their stimulation. Antibody production and binding to foreign antigens is often critical as a means of signaling other cells to engulf, kill, or remove that substance from the body. Lymphocytes destined to become B cells move to bone marrow instead of thymus to mature. After addition of cell receptors or antibodies, these are released into the blood. Once released, they move to the lymphoid tissue of the body. B cells play two important roles in providing the protection to the body. Antibody production against the appropriate target antigen, presenting antigen to T cells and therefore providing signals for T cell activation. Antibody production against the appropriate target antigen, then presenting antigens to T cells and therefore providing signals for T cell activation. The majority of B cell activation takes place in the lymph nodes. Certain types of cells in the lymph nodes eat anything foreign and present them to B and T cells. Any B cell that shares a receptor for its substance will be activated and start to multiply. B cells can also be activated by helper T cells. After activation, active B cells migrate around the body and change into plasma cells. So next we will be learning about plasma cells. Plasma cells are B cells that are responsible for the production and secretion of a single antibody type. This secretion gives rise to the antibodies found in the circulation. Immunity is kept for as long as the plasma cell continue to secrete antibodies. Then memory B cell. Memory B cells are also formed after stimulation. These cells migrate to the lymph nodes where they remain ready for further rounds of activation should be the same antigen ever be encountered again. Then T cells. T lymphocytes are usually divided into two major subsets that are functionally and phenotypically different. T helper cells, also called the CD4 plus T cells, are involved in coordination and regulation of immunological response. They function to mediate a response by the secretion of cytokines that stimulate B lymphocytes to secrete antibodies and TC cells to get activated in cytotoxic T lymphocytes, CTLs. The second type of T lymphocyte is cytotoxic T lymphocyte or CD8 plus T cells. These cells are involved in directly killing of tumor cells, virus-infected cells, or self-altered cells. CD8 plus T cells are also important in downregulation of immune response. Both types of T cells can be found throughout the body, most conspicuously in lymphoid organs, lymph nodes and spleens, but also the liver, lung, blood, and the intestinal tract. Lymphocyte CD8 cytotoxic T cells kill virus infected and damaged cells. Then CD4 helper T cells help cytotoxic T cells and B cells in their immune functions. Then B cells produce antibodies. Three types of lymphocytes involved in the immune response against infections against in the human body. Then natural killer cells. Natural killer cells are similar to CD8 plus T cells. They function as effector cells that directly kill certain tumors and virus infected cells. However, NK cells, unlike the CD8 plus cells, kill their target cells without need for recognition of antigen in association with MHC molecules. Monocytes. Monocytes are a type of white blood cells. They are the largest type of WBC and can differentiate into macrophage. Monocytes compose 2% to 10% of all leukocytes in the human body. As the monocyte begin to travel, they enter major organs such as liver and pancreas. As a part of the vertebrate innate immune system, monocytes also influence the process of adaptive immunity. Then next is about macrophages. Besides their role in phagocytosis, they may function as antigen-presenting cells or APCs because they ingest foreign materials and present the antigen to other cells of the immune system such as T cells and B cells. This is the first step in the initiation of immunological response. Macrophages stimulated by certain cytokines exhibit increased level of phagocytosis and also secrete cytokines that modulate immune response. Steps of macrophage ingesting a pathogen. 1, 2, 3, 4, 5. And just like that. Yeah, interpretation is given here. A. Ingestion through phagocytosis. A phagosome is formed. Look at this figure. Yeah, phago. Ingestion through phagocytosis. A phagosome is formed. The fusion of lysosomes with the phagosome creates a phagolysosome. The pathogen is broken down by enzymes. Waste material is expelled as assimilated. Parts: pathogen, phagosome, lysosome, waste material, cytoplasm, cell membrane. Yeah, that's how you need to interpret that figure. Various types of macrophages. Alveolar macrophage, splenic red pulp macrophage, marginal zone macrophage, peritoneal cavity macrophage, Langerhans cells. Dendritic cells. Dendritic cells also originate in the bone marrow and function as antigen-presenting cells. Dendritic cells are more efficient APC than macrophages. These cells are found in lymphoid organs like thymus, lymph nodes, and spleen, bloodstream, and other tissues of the body. They capture and process antigens in lymphoid organs where an immunological response is initiated. Dendritic cell in skin. Then neutrophils. Neutrophils are phagocytes that travel throughout the body. These cells are normally found in the bloodstream and are the most abundant type of phagocyte. During the acute phase of inflammation, neutrophils migrate towards the site of inflammation and are usually the first cells to arrive at the scene of infection. Neutrophils with segmented nuclei surrounded by erythrocytes. Intracellular granules are visible in the cytoplasm. Genes are stained. A phagocyte in action. Neutrophil engulfing anthrax bacteria. 5 micrometers. Basophils and eosinophils. Basophils and eosinophils are related to neutrophils. They secrete chemical mediators that are involved in defending against parasites and play a role in allergic reactions such as asthma. Eosinophil, basophil. Basophil and eosinophil. And mast cells. Mast cells reside in connective tissue and mucous membranes and regulate the inflammatory response. They are most often associated with allergy and anaphylaxis. Mast cells. So now let's have a summary. Both adaptive and innate response are carried out by a number of cell types. These cells are formed in blood marrow by the process of hematopoiesis. Following is a brief summary of various cell types involved in generation of immune response. B cell, B lymphocyte, a white blood cell that produces antibodies specific to the antigen. Basophil, a white blood cell that releases histamine, a substance involved in allergic reactions and that produces substances to attract other white blood cells, neutrophils and eosinophils, to a site of injury. Dendritic cell, a cell that is derived from white blood cells, resides in tissues and helps T cell recognize foreign antigens. Eosinophil, a white blood cell that kills bacteria that kills other foreign cells to big to ingest that may help immobilize and kill parasites, that participates in allergic reactions and that may help destroy cancer cells. Then helper T cells or Th cells, a white blood cell that helps B cell produce antibodies against foreign antigens, that helps killer T cells become active and that stimulate macrophages. Then killer T cells, a cell that attaches to infected cells and cancer cells and kills them. Then leukocyte, a white blood cell such as a monocyte, a neutrophil, an eosinophil, a basophil or a lymphocyte. Then lymphocyte, a white blood cell responsible for acquired specific immunity, including producing antibodies by B cells, distinguishing self from non-self by T cells, and killing infected cells and cancer cells by killer T cells. Then macrophage, a large cell that develops from a white blood cell called a monocyte that ingests bacteria and other foreign cells, that helps T cell identify microorganisms and other foreign substances and is normally present in the lungs, skin, liver and other tissues. Mast cell, a cell in the tissue that releases histamine and other substances involved in inflammatory and allergic reactions. Natural killer cell, a type of white blood cell that can recognize and kill abnormal cells such as certain infected cells and cancer cells without having to first learn that the cells are abnormal. Neutrophil, a white blood cell that ingests and kills bacteria and other foreign cells. Then phagocyte, a type of cell such as a neutrophil or macrophage that ingests and kills or destroys invading microorganisms, other cells and cell fragments. Then regulatory or suppressor T cell, a white blood cell that helps end an immune response. Then T cell, T lymphocyte, a white blood cell that is involved in acquired immunity and that may be one of 3 types: helper, killer and regulatory. So thank you for listening.